Hello, my name is Apollo Lee, and I'm a sophomore at High Technology High School. Today, we are going to be taking a look at my project, the aim of which being to strengthen the role of ginger, or Zingiber officinale, as an anti-neoplastic agent. Cancer is a huge problem that kills millions each year. Although current cancer treatment plans are effective, such as chemotherapy or radiation therapy, they do have their downfalls, with the first and foremost being cost. Asbestos.com states that over 63% of cancer patients reported significant financial struggles following a cancer diagnosis. Furthermore, these treatments can cause debilitating side effects, such as extreme nausea or immunodeficiency. So, is there a way to avoid the extreme route of treatment and possibly take some more bite-sized measures to help prevent it from happening in the first place, possibly in the form of a supplement? Let's take a look at ginger. Its rhizome, or root, has been used for thousands of years as both a spice and a folk medicine, and is pretty widely accessible to the general public. Studies have shown that the pungent derivatives of ginger exhibit the ability to modulate a wide range of signaling molecules and can either upregulate or downregulate gene expression to effectively restrict neoplasm growth. For example, 6-gingerol, which is a chemical compound found in fresh ginger, was shown to decrease expression of the cell cycle activators Cyclin A and CDK, while the chemical compound 6-shogaol was shown to decrease expression of MMPs, or matrix metallopeptidases. Now, these are enzymes that have been implicated as possible mediators of metastasis. So, what can we use to test the ginger? The answer lies in the yeast Saccharomyces cerevisiae. It can provide a reliable, biologically intact system in which to study the behavior of cancer cells because there's a very high degree of homology between the two at a molecular level. Approximately 30% of known genes involved in human diseases, such as cancer, also have yeast orthologs. And some of those orthologs are shown in this table, in which many of the cell cycle genes exhibited in humans also serve a similar function in yeast. For example, in these cancer-related pathways, we can see a similarity in, for example, the RAD50 gene found in both human and yeast. And this is responsible for the repair of DNA double-strand breaks. So based on this information, we can start to make an outline for our experiment. And so our objective, which I said already, we can also form an alternate and null hypothesis, both which denote whether or not the ginger treatment is effective. Finally, we have some basic assumptions here, where some include that the yeast growth won't be affected by any other factors, and that a lower proliferation of yeast indicates cell multiplication inhibition. Next, we have our list of materials for this experiment, which will be explained in our procedure. And now for our procedure. For this experiment in particular, it's pretty important that all safety precautions are taken. If the yeast is contaminated when incubated, it can cause a lot of problems down the line and can really skew their data. So doing things like using gloves and autoclaving all glassware are taken care of first. We start off by incubating our yeast in solid media using potato dextrose agar for 24 hours. Now this is done purely as a precautionary measure to make sure that the initial culture tube is not contaminated. Then once that was done, several yeast colonies from the plates were inoculated into liquid media YPD broth, and YPD stands for yeast extract peptone dextrose. The reason the transfer was made to liquid media is because ginger treatment can be applied more easily when it's mixed into liquid media rather than applied on top of solid media. So once that was applied, once I was incubated once more, the contents of the beaker were mixed and divided equally among 5 flasks. It was here that the concentrations of ginger powder extract were applied by breaking open the capsules in increasing weight volume concentrations of 0% up to 1%. So there's an outline of our procedure there, which I showed previously. And here are the flasks containing YPT broth with ginger concentrations just before incubation. Now, from here, we move on to the next part of the procedure, the goal of which being to analyze the impact that the ginger exposure had on the yeast. But there's a bit of a problem. At the moment, in the liquid we just incubated, there's way too much yeast in the media to, per drop, in order to count the yeast cells manually. So to solve that, we dilute the liquid by a factor of 100 by using distilled water. Now, this greatly decreases the concentration of yeast cells per drop of media, and also makes it feasible for the researcher to manually count the cells. So here's our implementation, and here are the conical tubes used to store that diluted substance. So even after you do this and make it so that there's less yeast cells per drop of media, it's still impossible to differentiate which cells are alive or died in the incubation period. So this was solved by using the dye Trypan Blue. Now this stains dead yeast cells dark blue by passing through their cell membranes, while alive cells remain untouched. You can see in this image, these yeast cells that are colored blue are dead. And here's our methodology again, and you can see here I transfer a little bit of the diluted solution into a microcentrifuge tube, and then I stain it with Trypan Blue. So now that there's a manageable amount of yeast to count and we know which ones are dead or alive, how are we able to obtain a reliable count of the amount of cells in a given space? Well, the compound microscopes we use certainly won't cut it due to the amount of inconsistency and unreliability in the zoom. So this was solved by using a tool called a hemocytometer. It's a thick glass microscope slide with two counting chambers, one here and one here. And these chambers are each laser etched with tiny grids, and this is what a grid looks like. So in this experiment, the amount of yeast cells found in these four quadrants, highlighted yellow, were counted, just for the sake of time. So the way we did this was by placing a special hemocytometer cover slide over the hemocytometer, and this creates a very tiny space in the chambers in which to put the liquid under. And then following that, we use a micropipette to place around 10 microliters of the stained 1% solution under this space. So here is where I take a bit of that stained solution. There's about 10 microliters here. And then you expel it in this tiny space between the glass cover slide. You can kind of see it there, and the hemocytometer. And you can see the liquid's kind of smushed there. So once that was done, you place it under a microscope, obviously. And then here, from examination, you can see, here's an example of a dead yeast cell. It's stained blue. And here is an alive yeast cell, along with two others here. This whole procedure was repeated for 12 trials each, and for the rest of the concentrations from 0.125% to 1%. So here is our raw data table, and here's our summative data table, along with our graph, which you can clearly see shows a line of best fit with a negative slope. From here, we have calculated the equation of the line here, and more importantly, through a linear regression test, we can calculate the p-value, and this will determine whether or not the difference, shown in here, is significant as the concentration increases. So by using a confidence interval of 95%, the p-value was calculated to be 0.043 approximately, and because this is less than 0.05, we can support the alternative hypothesis and reject the null. So based on this, we can make the conclusion that the negative relationship between the concentration of ginger and the proliferation of the yeast is negative and is significant. And here's just some of the regression statistics we used. We have our multiple R, R square, and we used all of that in order to get our p-value, which is highlighted here. So overall, this experiment did match my expectations, the results, and what this did was the role of ginger as an anti-neoplastic agent is greatly strengthened. So in the future, there are many ways to increase the integrity of this experiment and possibly take it further. For example, we can make use of mutant yeast strains with gene deletions that can make them more susceptible to the ginger treatment. For example, deletion of the ERG6 gene can increase membrane permeability and allow for more small molecule agents to enter the yeast more easily, such as ginger. We can also analyze other aspects of yeast other than proliferation. For example, we can measure changes in protein content that would indicate cell cycle inhibition as well. And another change we can make would be changing the model organism entirely. For example, Drosophila melanogaster, or the fruit fly, has also shown remarkable genetic similarity to human genes and would therefore be useful in testing treatments for cancer as well. Overall, I really enjoyed conducting this project, and I hope to continue with more research in the future. Thank you so much for listening.